fine will be somewhere in between. I think that's a fair statement. It'll be somewhere in between the two. There'll be some components of new applications of material that you've seen, but in a slightly different way, like the last question in the first midterm. And it's unlikely that you'll see questions that are as close to material that you've seen before on our final, but still hopefully familiar. So we're coming back to wrapping up the discussion that we've had about different kinds of bioreactors, how to run bioreactors in different ways. So of course we have talked about batch reactors. We talked about, what were the other kinds of reactors we talked about? Fed batch was the last one. So fed batch you do if you want high cell quantities, particularly if you've got some kind of substrate inhibition, or if metabolites interfere with the cells growing. Yep. Chemostat, so plain CSTR. Yep. Yep. So those are probably your most productive form, but are often not suited to many of the applications that we see, like without some modifications, not directly suited to some of the applications we see in bioreactors. What else? Yeah, so why would you use a multi-stage?
If you want to have different environmental conditions, you can have one tank with set A of conditions and then you move to set B of conditions and you move to a different tank.
Right, so if you maybe need to grow cells in a different circumstance than you're expressing the product, then you could have potentially more than one tank that allows you to have those different conditions, just like CC said. Is a multi-stage. There was one other that we talked about. For example, if you have very low substrate concentrations, like you might have to deal with in a wastewater plant. No. No. Fed batches. Fed batches when you want to, the high substrate concentrations will inhibit the growth of the cells. What do you do if you have very low substrate concentrations that you want to get even lower? Recycle, right? So that's when you try and put more recycle the cells back into the reactor. So even though you have very small amounts of substrate for them to grow on, meaning the growth rate of each individual cell will be quite low, by recycling the cells back in, you then can have a much smaller reactor that will effectively lower your very low substrate concentrations further, which is something you might need to do in a wastewater type system, where you're using what would otherwise be an environmental contaminant as the substrate. Okay, there was one other that we didn't talk about, and that's called a perfusion system. So this is a special system that is often used for animal cell culture. So, for example, you have we'll call it a bioreactor. But typically in this bioreactor, you'll have the cells somehow immobilized or recycled back in, and you'll have medium, and you'll have the medium goes in, and then you have product going out, along with dead cells and so on. This mechanism, perfusion, is actually often the way that people will keep cells alive in tissue engineered constructs. So if you're trying to grow a larger organ-like structure with cells in it, and you want to keep them alive by having lots of yummy nutrients come to them, it may be diffusionally difficult if you've got a larger structure to get that nutrient into the center of the structure without basically flowing the medium through it. So very commonly you see a perfusion-like system when you've got trying to grow a tissue engineered construct. In those cases, when you're growing a tissue engineered construct, you may not necessarily care about the product. What you're really caring about is the health of the cells. But in other cases, it may be a way of making your cells the happiest, and you then are keeping the cells and the cellular debris separate from the product. And if in fact the cells are making something in the bioreactor that's potentially inhibitory, you can see that it's being rapidly removed by having fresh medium come in and the old medium go out. So it's kind of like a CSTR, except your cells are staying in that system for longer periods of time. As well, if you've got dying cells and the dying cells are producing enzymes that can affect the cells or affect your product, they're getting separated out quite quickly, so you don't have to worry about it. Essentially, it's a constant growing environment for the cells, so they tend to stay happier. But what can you think about what might be some of the downsides of using a system like that? So it's generally a fairly gentle way of growing cells, because mammalian cells don't react nearly as well to these, high shear rate bioreactors that usually grow yeast cells or bacterial cells in. So it's gentler. But what might be a downside of a system like that? It may take longer. How much nutrient medium do you think you go through in a system like that? You end up going through a ton of medium. Because typically you can't wait until it's all used up by the cells, because that's defeating the purpose of removing the potentially toxic product and removing the cell debris and keeping it at a happy, comfortable. So it's not like in a CSTR, typically it's constantly grown at a relatively low substrate concentration, so you're constantly feeding medium in. Kind of like a CSTR, but in this case, you don't tend to have, you tend to run it at somewhat higher flow rates, because you've got the cells immobilized basically for returning to the reactor. You run it at slightly higher flow rates to keep the cells happy, but the result of that is that you're using a lot of medium, and your product can end up quite dilute. And as you know, the separations component of production is also incredibly expensive. But you do often get increased product quality and potentially reactor productivity. But then you also have lots of, along with the product, you have lots of waste. If you've got lots of medium going in, you've got lots of waste going out, and say it's a level 2 biohazard, you have to treat all of that waste as a level 2 biohazard, which can be not problematic, but can be expensive. Okay. But typically when you're growing larger structures in the lab, like on an experimental level, some sort of system like this is commonly used. So if you're, say, growing a tissue engineered blood vessel, typically it gets some sort of mechanical periodic strain to make it rearrange and make the collagen stronger, because the cells rearrange it when it's getting that, and you're also constantly flowing medium through it to make it happy, but you're using a lot of medium in the process. Okay, so those are our basic bioreactor ways of running the bioreactors that we talked about. Any questions about them now that we've sort of covered most of them? Okay, so what we'll talk about next is a little bit about what the reactors actually look like, how they're actually accomplish the air mixing that you need in order to make your cells happy. So we're back, not talking necessarily about mammalian cell growth, but probably reactors that are much more commonly used for bacterial cell growth and yeast cell growth. Ones like you've seen in the 4L lab and you will see in the 4L3 lab. Okay, so the three major types, going with big problems that you have in running a bioreactor is the sort of the two big problems you have. One is getting enough oxygen to your cells, and the other is getting enough heat away from the cells. So most of the reactor designs are designed with the view of getting enough oxygen to the cells. So internal mechanical agitation, that's talking about ones that have impellers or turbines in them. So that's like the bioreactor that if you had the opportunity to deal with the bioreactor that you have seen in 4L2, will see in 4L3. How many people actually did have that opportunity? So, a third of most of you will have that opportunity. Being in the bio program, you'll get to do it next term. So, basically it's got like a little fan type deal that stirs it up. Okay. Bubble columns, I'm going to go into this in a little bit more detail. Bubble columns basically use the bubbles to mix the reactor. And then a loop reactor is sort of a bit of a combination of potentially the bubble and the internal mechanical agitation, but it sends the medium and the cells around in a loop, but using the basic principles above. So, and bubble columns typically are a bit taller than the internal mechanical agitations. So this one is the most common. Okay. So why would we have, what's the point of having that stirring? What does it do for you? Big agitator is it going around?
More surface area?
For what?
So you're breaking up the yeast not from like for example, to like actually run your reaction. So smaller bubbles means more surface area.
Okay, so more surface area on bubbles. That's true. It as you're stirring it, your bubbles break up into smaller bubbles. So even though you're sparging the air from the bottom, but the mixing shears the bubbles so they become smaller. And that's really helpful and important.
You increase mass transfer?
Pardon me?
You increase the mass transfer?
It does. So it does that in a couple of ways. Why would it increase the mass transfer?
The velocity.
So, yeah, basically you'd have a smaller stagnant film. Yeah.
It increases the likelihood that oxygen will be leaving the cells.
Yeah, well, same, yeah, both those things are true. So you've got higher velocity, but I'm just going to hold off on that. But the other thing that happens, think of the path that a bubble takes from the bottom of the sparger. If there's no agitation, it goes straight up. What does it do if there is agitation? It takes a long time for that bubble now to get to the surface. So you're increasing not just the mass transfer rate, but the time that it can happen through. So increased mass transfer rate and time for the bubbles. Okay, because so the large bubbles are sheared to form smaller bubbles, so you've got increased mass transfer because you've got increased surface area over which that happens. They're moving around faster, so you've got less distance for the air to diffuse through, and they're there for a longer time. The other thing, of course, that it does is if you've got a small departure right at the base of the reactor, what'll typically happen is the bubbles sort of rise like that, and you end up with some parts of the reactor that aren't as exposed to the air. So if you're mixing it, you're mixing the air through, but you're also mixing the organism through, whatever it is, so that everything's getting a more even interaction with the medium and with the air and so on. So, just better mixing of both the air and the organism. Right? Yeah.
So out of curiosity, just, is there a maximum mixing? So is there a maximum that it can, so reach a certain limit and it would shear too much that would
Yeah, so there are a couple of problems with mixing too much. So you might think, you know, you have lots and lots of air, let's just bubble the heck out of it and spin the heck out of it, and we'll have happy organisms that make lots of product. But Eva's absolutely right. If you have too high a shear rate, all cells are vulnerable or shear sensitive to some extent. So E. coli is pretty sturdy, but it's still possible to mix it too fast and basically bust it open. Same thing with yeast. Mammalian cells are incredibly shear sensitive, so typically you cannot grow them in a bioreactor like this because you start putting bubbles through and spinning them around and you'll get a really high rate of cell death and very low production rate because they don't have cell walls to protect them. So problems, but by shear. Okay, so high shear can damage the cells. Can you think what else might be an issue if you have a lot of bubbles going through and a really high mixing rate? What happens, let me think about an example that would be an everyday example. Hard to think of it. One of the big problems, actually industrially, that isn't necessarily obvious and apparent is foaming. So the organisms make things that stabilize small bubbles, so basically surfactants, and they release those. And it's a really big problem in bioreactors that you get basically a head on your bioreactor. So you pour beer and you get that big pile of bubbles. Well, the same exact thing happens in bioreactors. So if you're there are components in the medium because of the cells that will stabilize these small bubbles. So if you're putting a ton of bubbles in and mixing the heck out of it, suddenly your 3/4 full bioreactor has foam coming out everywhere. And it makes a giant mess and you lose sterility, and it's a big operational problem in running a bioreactor. And so one of the things that you're going to do even in 4L3 is you're going to put a little bit of anti-foam in it, which basically breaks down the bubbles. It's like putting a little bit of grease in it. Has anyone done the trick where you've got the head on your beer and you try and get grease from your nose and stir it up and it breaks the head? So anti-foam is sort of similar. It's like acting against the surfactants that the cells are releasing. It decreases the problem of foam, but obviously you don't want to add lots and lots of chemicals that may not actually be good for your cells either into the reaction system. So the combination of how you run your reactor and what your cells are doing and how much anti-foam you add can help deal with that issue of foaming. But it is a problematic thing. So any questions about that? So the other thing to note is, with any bioreactor, usually because of that foaming problem, you have to run it at less than 3/4 capacity. So if you have a 2-liter bioreactor, you don't really fill it to like the very, very top, because then giant mess everywhere. And I mean, I think the same thing maybe is true in brewing beer. You don't fill it to the very, very top.
Yeah, when we're making beer, I mean, for a 5-gallon batch, we maybe start with 7 gallons and boil it down, and we use 10-gallon water, so.
Yeah.
3/4.
Yeah. Yeah. So you especially because of this. I mean, you're spinning it, and so there's some creep up the walls. But in particular, the foaming issue means you have to have a fair bit of headspace left to run your reactor successfully. Okay, and then the next thing is, how do you actually, the air is supplied by a sparger. So how do you actually do the mixing? So you've got air coming up, and there are 2 basic impeller types. The most common one is called a Rushton impeller. So a Rushton impeller, if you look at it from the top, and Ryan, if you want to actually, the pictures are quite nice in the notes. Maybe just pull them straight from the notes. Let me know if you can't pull them, I can pull them for you.
Oh, I can do a screenshot.
So,
What lecture slide are we on right now?
that is a very good question. We're on lecture 16, lecture slide 5.
Oh, nice.
Yeah, see them? So if you want to look in the book and in the notes, the pictures are much better than I can draw. But that's what it looks like from above. Basically, I'll demonstrate it better. The blades there are 6 blades. They're straight out. They're like paddles, and it just spins like that. So as you can imagine, what happens to I'm not going to do it anymore because I'm going to get dizzy. What happens to the liquid is it just spins around like this, right? And so those are really common. You see them almost all the time in lab scale bioreactors still. You still see them in a lot of industrial scale bioreactors, but it has been recognized that they're not the best impellers to use because they cause a lot of shear stress. Okay, so when you're in the reactor itself, it just sort of swirls the liquid around like that. But you don't have really any mixing top to bottom. You have really good mixing around and around and around, but not up and down. So instead, what they did is came up with the other main impeller type is called an axial flow hydrofoil impeller. Typically they'll have 4 paddles, and basically all they did was take the paddles that were facing straight and turn them. So that now you spin around and you're no longer just pushing the liquid around and around and around in a circle because it's tilted. What's it doing? Yeah, it's now mixing up and down as well as around and around, right? So those have lower shear stresses to the cells, and better vertical mixing. they actually, because of the better vertical mixing, they actually have better oxygen transfer as well. As a consequence. I would say it's just historical reasons that Russian impellers are still used so often in lab scale and industrial processes. The axial flow hydrofoil impellers are more effective. So if you're speccing out a new one,
So that's
this is the one that's tilted. Okay, so the next one on my list was bubble columns. So here, basically, you've got your sparger at the bottom that has lots of little holes in it so the air can come through, okay? And the air goes, and often, oh, something I forgot to say, in both of these cases, in the reactor itself, they often have baffles added that increase the mixing as well. So there's a combination of an impeller and some baffles in the reactor that increase the turbulence of the mixing. In the bubble flow ones, you actually have, so there's your inlet, and then you often have sort of perforated plates partway through. And so the bubbles go up, they hit the perforated plates, there's some mixing, and some of the bubbles go through, they hit the perforated plates, there's more mixing, and then the air, the gas comes out the top. Okay, so it's the perforated plates that really enhance the gas dispersion and the mixing. So typically these are relatively, I mean, as you might imagine, really in this case you've got bubbles coming up and then they're being mixed around radically. These ones you've got bubbles coming up and relatively gently moving through the reactor. So there is considerably less shear associated with the bubble columns. They're also energy efficient because you don't have to put lots of energy into spinning things around. You're basically just sparging the air, and the air is doing most of the work for you. It's just how the design is that you have mixing. And it typically is actually, it's cheaper because it's energy efficient. And you have a little bit less places that contaminants can come in because you don't have the impeller system going as well. So that I wouldn't say that's a huge advantage. This is, I would say, a problem common to both the, like really any system where you've got bubbles coming in, but cells like bubbles, and bubbles bursting is bad. So as if you've got lots and lots of bubbles coming up, the cells will actually tend to stick to the bubbles, and this is part of the problem with foaming actually. In the foam you can have a really high amount of cells. So if you've got lots of bubbles flowing around in here, you can have cells accumulate on the bubbles, and especially if they burst as they go through the perforated plates, you can end up damaging the cells. So there's some trade-offs to using a bubble column. And they're also not as effective at, I mean, you can imagine if you have lower flows and lower shear stresses, you're probably also getting less mass transfer from your drug cells. So the sort of compromise is a loop bioreactor. So there are some that actually just use the airflow, sort of just a slightly different concept than that. They basically are bubble reactors with a loop in them, and some actually use a pump system. So again, if you look in the notes, they're a little bit prettier than what I'm going to draw, but in one setup you have your inlet. So you've got basically a cylinder that's open at the top and the bottom within the bioreactor. And the inlet air, this is called an airlift loop. The inlet air goes up the middle of the cylinder, and then the gas leaves, and then the rest of the medium goes, so as the air goes up the middle of the cylinder, it brings the cells and the medium with it, and as the gas leaves, they drop back down, and they're cycled around and around the loop in that system, right? Opposite way also works. So this is called a propeller loop. And really it's the same system, but here we've got the air inlet going on the outside of the cylinder. Okay, so then it takes everything up there, and all the cells and medium go back down the center. Okay, there you often have, to ensure that it goes back down the center, you often have a motor with a little impeller that's driving the fluid down the center of the reactor as well. Okay. So in that sense, in this one, you probably have a little bit of shear stress than in the straight airlift loop bioreactor. And then the final one I'm going to show you is called a jet. And it's essentially the same thing. We've got the cylinder again. But the difference is we've got air into the center. So again, we've got the same pattern as in the airlift loop. But there's also a medium recirculation loop that is jetting the air out with some extra medium into the center. So it's not just driven by, it's not just a passive buoyancy of the air bubbles that's driving the flow up the center of the column, but a mixture of air and water actually being pumped up the center with a little bit more force and speed. So again, you have a little bit more, you have more driving force for oxygen transfer, you have a little bit more shear stress as well in the jet bioreactor. Okay. So airlift bioreactors in particular tend to be for very large fermentation. So even greater than 200,000 liters. Because it's very difficult to achieve mixing, to scale up mixing with impellers, even with a just straight bubble bioreactor. But the airlift or the airlift loop bioreactors actually can be quite easily scaled up, and you can manage still really remarkably good mixing in very, very large systems. So if you're looking for something that's really, really large scale, the airlift loop is commonly used. I think it says the largest airlift loop bioreactor is 1.5 million liters. So like really substantially large bioreactors.
It's swimming pool.
Yeah, you can get some really big ones. And they're the reason that they're so effective is because you can get higher oxygen transfer rates using these methods without adding unreasonable amounts of shear stress. So when we talk about scaling up, we'll start seeing how shear stress translates when you suddenly have to do.
You're supposed to write a midterm right now.
I think Ryan has a midterm that I hope you're not missing.
Yeah, 5 minutes ago.
Oh no. So obviously I'm such an engaging speaker.
I really thought it was 5 o'clock for some reason.
Oh my goodness. Yeah, even
I get to watch this on video later, too.
At 5:30, and you can watch it again and again as you come to write out your notes for the rest of it. So good luck on the midterm.
Thanks,
I'm sure
I'm
it'll
going
go
for a little
well.
run right now.
You know, they say exercise is good stress management.
Or increases your stress if you're 5 minutes late. Not my bad.
Good luck.
Thank
Good
you.
luck. That's one of those like nightmare moments. Like, you know, you dream about moments like that.
You're fine.
You know, it's gonna be time to tell you where you go. I think it's for finance business. For part finance. Okay. That was scary.
When the other class will write it, it comes out.
Just a lot. So if you were making a bioreactor, what would you make it out of?
Jelly beans.
Jelly beans. Interesting idea. The jelly beans can provide substrate as well as they slowly dissolve into the medium. Keep up the high substrate quantities. But what would you make? Other than jelly beans, what might you make a bioreactor out of? The vessel, the actual reaction vessel. So metals, so stainless steel. Why did you say stainless?
So that's good steam. So you put oxygen into it, and also for sanitation purposes. If you have to like, you're dealing with bio stuff, you have to like have caustic or something.
Yeah, so you want something that's not going to corrode. So you don't want like plain iron that would obviously be throwing lots of rust into your system. In fact, growing organisms in general is really difficult as far as corrosion goes. Yes?
Actually, I have a question.
Yeah.
What would the benefits of using glass be, besides visibility?
So glass is typically used, so that's the other option really. Glass is typically used for small bioreactors. Just structurally, it's not strong enough to go big. Anything above 50 liters, so I'll just write that down. So below 50 liters, you can use glass. It's just not structurally strong enough to withstand the processes that it would have to go through and hold the amount of liquid that it would have to hold above 50 liters. But it is really nice because you can see through it. And that's really the reason that you use it. So it's really nice to be able to see the cells grow, and it's completely corrosion proof. So glass will not be, you can keep it forever and it's not going to corrode because you have cells growing in it in a high oxygen environment. And corrosion is actually a big, big problem anywhere that cells grow. So cells actually will have the potential to cause, they grow on a surface. Cells really like growing on surfaces, especially bacteria. So in nature, in fact, most often bacterial cells aren't growing in suspension, they're growing on surfaces. So like your teeth, for example, or the slippery rock in the river. Bacteria really like growing on surfaces, and you're providing them with some really nice surfaces to grow on in these reactors. So when they're growing on those surfaces, they actually can provide little redox reaction points that can cause corrosion pitting. It's a big problem actually with shipping. It's a big problem in pipelines. It's a big problem in the food industry is you have all of this corrosion pitting caused by organisms growing on metal and having redox reactions. So stainless steel is very helpful. So you'll see anything that is in the food industry, anything that is in the bioreactor industry that has cells growing is stainless steel because the cells can damage it. And also, as Carl pointed out, you need to clean it and sterilize it, and the harsh caustics also are really difficult on the system. So, yeah, it's actually typically not just stainless steel, but 16 stainless steel. So you can use a slightly less corrosion resistant stainless with like the tops of the bioreactors that aren't going to come in direct contact with the cells. 304 stainless steel, which is a little bit cheaper.
So price-wise, glass would be more expensive, right?
Glass is typically more expensive, but also glass is typically what you see in lab scale bioreactors because it's really, the ability to see the cells growing is very, very helpful at the lab scale. But industrially, glass is typically not used. But it's really nice for, you know, in the lab, you peel back your cooling jacket and take a peek at it. Oh, it's completely clear. That's not a good thing. So that happened actually to one of the labs last year. We talked about plasmids and how when you genetically engineer an organism, typically you put some sort of selectable marker on it. So it's, for example, resistant to antibiotics, and as long as so then you can put antibiotics in your system, and all the non-genetically engineered organisms or ones that have kicked out the plasmid won't grow or will grow very, very slowly compared to your genetically engineered ones. Well, we're pretty sure that one group picked cells that were didn't have the plasmid in it and didn't have the genetically engineered protein in it at all, and then put their, so what happened was we had two genetically engineered E. coli's, one to produce one protein, one to produce another, and they had different selectable markers on them. And we were pretty sure that the one group picked the wrong one and then used the selection medium for the other one. So they basically grew up their cells and then killed them all by adding antibiotic. So they, 2 hours later, they peeled back their thing and looked at it. I mean, they were doing their measurements, and it basically was their measurements of optical density were basically telling them that there were no cells there. But if you look at it, it was clear there were no cells growing in their bioreactor. So it's a good way of just doing a quick visual peek to see what's going on, and you can see what your sparging looks like and your agitation rate looks like and all of that sort of thing. So it is nice to have the ability to look at your bioreactor, and it is nice because it's corrosion resistant. But of course it's quite delicate as well, so you don't want to drop your bioreactor as you're bringing it to the autoclave to autoclave the whole thing and sterilize the whole thing. Be very careful, please. But yeah, typically glass is really nice because you get less biofilm formation, you don't have corrosion, and you can see through it, but it's just not structurally sound, and it's more expensive. The other big things to consider, of course, you have to, with large, large mechanically agitated bioreactors, height to diameter ratio is usually about 2 to 3. So it's sort of 2 to 3 times taller than it is wide in the very large bioreactors. But other things you also have to be concerned about are having sterile air inlet, sparger, baffle plates and impellers. Cooling coils. So in very large reactors, it's not enough to have a water jacket or even a glycerol jacket around it to cool it down. You have to have internal cooling coils because otherwise you don't make it up. As well as having chemical anti-foam, you can actually have a mechanical foam breaker at the top. And we talked about the working liquid volume of being less than 75% of the total volume of the reactor. Typically as well, you'll install steam lines so that you can do sterilization in place, because the large reactors, you can't walk them over to an autoclave. So you'll do caustic cleaning, and then you'll rinse it, and then you'll basically put really high temperature steam in it for a certain period of time that ideally will sterilize the entire thing in place. But the cooling coils in particular illustrate the problem of heat transfer. So I told you that oxygen transfer and heat transfer were the two major limiters. So large bioreactors may need internal coils rather than external jacket. Having said that, it really depends on your system. So touring the Labatt plant, for instance, they have really large batch bioreactors that brew up huge amounts of yeast fermented beer at a time. They just have external glycerol jackets that do the cooling. And the reason you might choose internal or external glycerol jackets is because then you don't have to worry about cleaning and sterilizing all of those little nooks and crannies that you do with, and it's just less maintenance as well. It's easy maintenance. Yeah.
Another thing is they usually, if they do use coils, it's usually the cold water goes through the coils instead of the beer going through the coils, cold water, because it's also easier.
Yeah, yeah, so they definitely, the heat transfer is from coils to the fermentation broth rather than running fermentation. And that's also because if you have relatively small coils with things growing in them, you get biofilms on the surface which can clog the coils. And in fact, one of the issues there with the coils as well as with the external jackets is that biofilms can reduce heat transfer. So if you end up with large, relatively thick biofilms growing on the surface of the coils, your heat transfer slows down, so you have less effective heat loss, which is not ideal. Just to quickly, before the end of class, go through the other problems. We talked about foaming. So if the foam escapes, it can wreck the air filters. It blocks air filters. You'll see in 4LL, as soon as you have your HEPA filter that gets wet, it no longer works, so there are little HEPA filters on your pipetters. And if you make a mistake and pipette too high up, your HEPA filter gets wet and no longer your pipetter stops working. Same thing, your HEPA filters won't work if they get wet. So you typically don't run them full, and you have mechanical breakers that sort of chop it up, and you have chemical things. We've also talked about sterility. So you have to make sure that all the inputs are sterile and that you can actually sterilize ideally in place. And cleaning is the other big issue. So if you can clean it in place by having feed lines into it that run your caustic into it, hot caustic into it typically, that's how they're cleaned in place, and then you rinse it out and then you sterilize it. So those are all considerations you have to keep in mind with the bioreactor system. What we're going to start talking about next class is oxygen transfer.